Hello, my name is David Earl Hostallero. I am a PhD student at the IMAD lab in McGill, and I will be presenting the study we conducted with our colleagues in Mayo Clinic, supervised by Professor Amin Imad. The research is called a deep learning model of preclinical to clinical anti-cancer drug response prediction and biomarker identification. In precision medicine, two of the major tasks are the following: prediction of clinical drug response or CDR, and the identification of biomarkers of drug response. In the prediction of CDR, the problem formulation is on the left. Given the molecular profile of a patient tumor, how would the tumor respond to a drug? The problem is tightly knit with biomarker identification on the right, where we would like to find out which genes significantly affect the tumor's response to a drug. Typically, we would train a model to address these tasks. We are specifically interested in deep learning because it has seen many success in modeling complex relationships. However, clinical drug response data is so scarce that training and testing on clinical data alone is very challenging. One could say that we could use preclinical data or in vitro drug response data to train our model. However, models trained on preclinical data typically do not translate to clinical data because of various biological and statistical discrepancies. These discrepancies can be seen by simply plotting the first two principal components of tumors, here in orange, and cell lines here in purple. Additionally, deep learning models are often regarded as black boxes. In this study, we would like to open this black box to identify potential biomarkers of drug response. We propose a deep learning pipeline called Tyndall to address these two tasks. Our pipeline is composed of two phases. The first phase is concerned with CDR prediction. Here we train our deep learning model using the gene expressions of preclinical cell lines and their log IC50s for a specific drug. We then use our tissue-informed normalization to match the distributions of tumors and cell lines, therefore allowing our trained predictor to successfully predict for tumors. In phase 2, we use a neural network explainer to calculate gene contribution scores of each tumor samples. The scores are then aggregated and filtered for further studies. The tissue-informed normalization is a simple yet effective method to match the distributions between cell lines and tumor gene expressions. First, we identify the types of cancer or tissues for which a drug is typically prescribed. For example, we identified cancers in breast and soft tissue to be the main target tissue of tamoxifen. Using unlabeled breast and soft tissue tumors, or tumor samples, meaning that they are not part of the test set, we calculate the means and the standard deviations of their gene expressions and use them as z-scoring constants mu and sigma for the testing set. This allows the model that is trained on preclinical samples to translate to this tumor domain, guided by the target tissue and cancer types. In the second phase of the pipeline, we use a black box explainer called C-explain to give feature attribution scores for each gene. This explainer is actually another model that is trained by comparing the errors of the models when features, or in our case genes, are removed one at a time. This is a reminiscent of Granger's causality. In our study, we use cancer cell lines from GDSC and patient tumors from TCGA project. We only evaluated on 14 drugs that are common between the two databases and have sufficient number of testing samples. We used one-sided Mann-Whitney U-test to check if our model could distinguish between sensitive and resistant patients. We observed that our simple model segregates the classes in 10 out of the 14 drugs, which actually outperform several other models and baselines. Furthermore, we introduced a metric called precision at kth percentile, where a high value means that patients with predictions within the kth percentile of the distribution corresponds to the true sensitive patients. Tyndall can identify responders with a precision at kth percentile above 84% for any value of k. Comparing against various models and baselines, one result we wanted to highlight is the analysis of the embeddings of preclinical and clinical samples. On the left side is the comparison of embedding distances per drug compared to domain adaptation-based methods, DAN and ADA, and batch effect removal method called COMBAT. Having points below the diagonal means that the distance for the drug is larger than Tyndall's for the method in the x-axis. We can also see on the right the first two principal components of the latent space embeddings, where Tyndall displays a nice overlap between the two domains. Using the neural network explainer of Tyndall, we identified 10 candidate genes that significantly contributed to the prediction of Tyndall for one of the drugs, tamoxifen. We then used siRNA knockdown experiments in two breast cancer cell lines, MCF7 and T47D, to see whether the knockdown of these genes significantly affects their response to tamoxifen. Our experiments confirmed that tamoxifen sensitivity is significantly influenced by all of these genes for MCF7 and 7 of these genes for T47D cells. In summary, we proposed Tyndall, a deep learning pipeline for preclinical to clinical drug response prediction and biomarker identification. Our model could successfully distinguish between sensitive and resistant patients in 10 out of 14 drugs, outperforming various models. The embedding distributions between preclinical and clinical data are better matched with Tyndall, and we identified potential biomarkers of drug response. Finally, we experimentally validated 7 genes identified for tamoxifen in 2 cell lines. If you want to learn more, come visit our poster during the poster session or read the paper link below. Thank you.